quite a few up here. Come on down. All right, 6 o'clock. Okay, welcome everyone. It's finally 5 degrees warmer in here with 100 or so biological heaters going. All right. Welcome to the first seminar of the Spring Science and Engineering Talk. My name is Adelisa Flores. I teach chemistry here. I've been part of the planning team for about 5 or 6 years now. And I want to give just a brief history in terms of how it got started as far as bringing all these great speakers and great research scientists and engineers from the laboratory. And we have Nan Ho, our dean, to thank for that. She's over there. Some of you probably know her. She was a former biology faculty, and she spent her sabbatical at the lab, and she got to go to these really great seminars, right? Interesting seminars, and she wanted to bring that over to the campus, and that's why we have this. So this is our 8th year. We're finishing up our 8th year. And so this is actually our 31st seminar. So if you've been here a while, you've seen some of these. So before we start, I want to thank, of course, from the lab, the speakers. I will be introducing them in a couple minutes here, but also I want to acknowledge the people who in the lab who've been helping us bring these wonderful speakers over. One of them is actually here, Joanna Albala. Where are you? She's right there in the back there. She and Chris Culp from the lab have been really, really helpful trying to invite these great speakers so they can come talk to the students, really, right, to bring all this good stories about research that's being done over there. Chris Culp is the group leader in the Biosciences and Biotechnology Division, and then Joanna Albala is the Education Outreach Manager in the Science and Technology Education Program. On the LPC side, I want to acknowledge the faculty who encourage their students to come, and also those who come. I see quite a few faculty over here. It's really great to see that. Also, we've had classified professionals and administrators come forward. I don't see anyone, however, so I'm going to keep continuing on here. Okay, so the last set of people I want to acknowledge, this is really a team work. It takes quite a bit of work to kind of put these together, right, for this one night. So I want to acknowledge all the people who have been helping out. First of all, I want to say a big thank you to Carol Schill. She's our new STEM coordinator. She's over there. Some of you probably recognize her. She works with the engineering tech as well. David Lee is the IT guy who's been helping set up all the nice audiovisual technology that we get to use up here. And of course, I want to thank our dean, Nan Ho, and her. She's still going, even though she's now our dean, and Linda Cross in the division office, right? So they've been really helpful. Thank you. Excellent. The other people I want to acknowledge are the faculty planning team. We have Mike Ansel over there. I see him over there. He's Robin Rehagen in physics. I know she couldn't come today. You guys know who I'm talking about. Barbara Zink from biology, of course. And Keith Level from engineering. So those are the people in the planning team. And lastly, I want to acknowledge the people who came before us. Dr. Neil Eli, our former dean. Dr. Lisa Everett, another former dean, who've been as helpful as Nan in getting this set up. And also from the lab, Dr. Beth Vitalis and Dick Farnsworth, who were the part of the original team that brought this together. So now on to our seminar, right? Because that's what you guys came here for. Bioprinting, bringing life to 3D printing. So you get to hear how biology, engineering, and medicine intersect, right? To bring us this cutting edge work, right? That Dr. Monica Moya and Javier will be talking about. So real quickly, Dr. Monica Moya is a biomedical engineer. So you got all that biomedical engineer in there. Researcher in the Materials Engineering Division at the lab, Lawrence Livermore National Lab. She works as the principal investigator and technical lead on 2 bioengineering projects. And her research interests, as you can see, is 3D bioprinting, organ on a chip, sounds interesting, and integrating engineering and biology. So that would probably come up quite a bit today. Her Bachelor's of Science is from Northwestern University, and her PhD is in Biomedical Engineering from Illinois Institute of Technology. And she's written a lot of publications. Javier Alvarado, right there. He currently works as an engineering tech at the lab, contributing to several bioengineering projects. He got his degree in Bachelor of Science in Neuroscience and Behavior at UC Santa Cruz. Some of you may end up there. And later he developed his molecular biology skills by earning a biotech certificate at Ohlone College. And he also has taken classes at LPC, right? Engineering classes, so he's probably going to talk a little bit about that. Because he was interested in learning engineering principles and their application to biological systems, right? His research interests include 3D cell culture, biomaterials, and applying 3D bioprinting techniques to model in vitro the neurovascular environment. So without further ado, let's give him a big hand.
Thank you. Okay. Sorry, I was told to definitely keep it close, so might have went a little bit overboard with that. Okay, thank you for that introduction. Javier and I are going to split up the talk, and I will start things off and then take it over to Javier. Okay, so let's start. So this is maybe what you guys think about when you think of bioprinting, right? You think, oh, that must mean that they're printing something that is alive, like a heart. And certainly this is, I guess, sort of the big goal and promise of 3D bioprinting, but we're not quite there yet. And what makes bioprinting particularly a really exciting tool is that it really starts to bring engineering principles to biology. So as opposed to just being able to passively observe biology, 3D printing allows us to manipulate biology and to be able to engineer it in different ways. The field of 3D printing is definitely not new. It has been around since the early 1990s, but its application to tissue engineering, as you can see up in this chart, has been around for maybe the last decade or so. And a lot of the original, I guess, examples of using 3D bioprinting, a lot of it was involved in the technology development and printing very simple tissues. So here's a couple examples of some of these simple tissues that people have printed. They're pretty cool, although I like to call them boring organs because they're not very complex organs, right? So you've got, for example, from Wake Forest, a bladder, somebody 3D printed an ear and a trachea, and also skin. And with the exception of the skin example, all of these first started off with just printing the scaffold, meaning that the structure was printed first, and then they seeded the cells onto it, which is a great way to start off with 3D bioprinting because then you don't have to worry about keeping things alive. Certainly, though, when you're not printing with cells, you're losing the ability to place them in certain locations. So the skin example definitely used cells, but the other thing is that it's a pretty simple organ, the tissue, the skin, right? Like it's a organ that's pretty thin. And that's the other thing about most of these organs is that they're either thin, if you think about bladder or skin, or they don't require a lot of vascularization. So cartilage doesn't require a lot of vascularization. And the reason I even mention vascularization is because it's really kind of the plumbing system of our body, right? So if you want to build something, I'll use the analogy of a house, you have to include a plumbing system. Otherwise, you're not going to be able to inhabit it for very long. And it's actually a pretty overgross simplification when I say that the plumbing, that our circulation is like the plumbing system, because oftentimes our circulation is really tied into the function of our organs. So for example, the liver, right? We know that the liver filters. Well, all of that filtration happens through the vasculature. And when you think about, too, the differences between the vasculature in the liver or in the lung or in the brain, it also tends to vary. So again, going back to the liver example, the vasculature there is incredibly leaky. However, if you think about the vasculature in your brain, thankfully it's not leaky. It's very exclusive. It only lets certain things in. Luckily, one of those things happens to be coffee, so that's great news for us, but it doesn't let other things through. And so the problem is, if I'm going to decide I'm going to make vasculature, the thing is, not all vasculature is created equal, right? Like even from these pictures, you can tell that they look really different. And so if we really want to start to create vasculature, we have to keep that in mind, that we're going to have, that a one-size-fits-all approach is just not going to work. And the reason, again, that I said that we want this vasculature is because if we really want to go up in 3D space, really create larger tissues that are going to need more than just simple diffusion to keep them alive, that vasculature piece becomes really critical. So increase complexity, you have to increase also your vasculature. So I know I threw a whole lot at you, and you probably have all sorts of questions, and you're like, wait, what? You're printing? Like, what are you printing? Why? How? Where? Hopefully by the end of this talk, we're going to answer all of these little thought bubbles. So I'll start off with one of the really easy ones, which is who? Meet the team. You'll be hearing from this guy, Javier, pretty soon. We have permanent team members are Javier. I also have a postdoc named Rick, so a lot of the work that you'll see here is work that Rick did. And then Joanne, who is a biotechnician. Then we also have a lot of different people that have come through as interns, and they vary from anywhere from, actually last year had a high school student all the way up to grad students, and actually also a science teacher. So the point is that we do a lot of research with people of all different levels, people that are undergrads, grads. Sierra, who is in the back, and I'm going to call out and embarrass her, is currently a student at Las Positas, and she's been helping us out. I think we have a couple of her images in the talk. Yeah, so you get to see some of the cool stuff that she's been helping us do also. Okay. Okay, so what is it exactly that we are bioprinting? So our bioprinting approach, I like to joke around and call it the Bay Area kind of approach, because it's one that's kind of organic-centric, meaning when I'm thinking about bioprinting something, I want to give some structure to it, but I don't want to dictate all of the structure. And the reason for this is because biology is very complicated, right? And if I have to try to replicate all the pieces of biology, it's going to be really hard. And so I don't try to put in a whole lot. I mean, I try to put in minimal structure and then also let the cells help out. So in a way, I'm kind of co-engineering with nature, right? Because if you think about it, our cells are the original engineers of life. Inside of them are the little blueprints, and so they know how to do the things that, you know, they were programmed to do. And so what I do is I create a minimal structure, make sure that I have a material that lets the cells do what they naturally are going to do, and allow them to self-assemble. So the two examples that I'm showing are two ways that you can get vasculature to form. One is I can create tubes, which is what you're seeing on the top, and the other one is I can create a bioink that makes the cells make the tubes. So here's a artist rendition of the process. It's a little bit cheesy. It has a MakerBot, which was the first the first 3D printer that I used to start bioprinting, but we now have a much fancier system with better resolution. But essentially, we use the printer to give us a sort of X, Y, Z control. And so what you see that is being extruded out is one type of ink where the cells are sort of dispersed, so they're not in any particular geometry, but you see that we did put some tubes and some structure. The reason we do this is because we want to be able to tap into the biology, meaning we want to be able to introduce fluid, because that's what vasculature has, right? There's stuff moving through it. And because we can introduce fluid, we can control gradients, and we can motivate these cells to grow in a certain direction. So if you can think about it, we can introduce media that has nutrients in one line, the other line we don't put anything, and of course the cells want to move up towards the area that has the nutrients. So people call it 3D printing, but I always say you might as well call it 4D printing, because it turns out that the fourth dimension, which is time, ends up being absolutely critical when you're bioprinting, mainly because you have to keep cells alive. You have to keep them alive as they're waiting to be printed, then you have to keep them alive as they're printing, and then the most important part is keeping them alive after you've printed them. And so this is what we call the post-print process. And this is a key distinction between regular 3D printing versus bioprinting. So in 3D printing, you print whatever it is that you're trying to print, and at the end of your print process, you have something that you can readily use, right? Maybe you printed a tool, or you printed, I don't know, some kind of an object that's ready to use. The problem with 3D not the problem, but the challenge with bioprinting is that when you print something, even under the best scenario, you still have the beginning of a product. You still have to continue to develop it because it's something that's alive. And so this fourth dimension of time becomes really important. Okay, so back to what exactly are you printing? Well, we have a variety of different types of inks, and so we have a toolkit that we pull from, and so it depends on the application what kind of ink we're going to use. So I'm going to walk through some of the different types of inks that we have. So I talked about this one, which is the co-engineering with nature sort of approach. So for these inks, what we're doing is making sure that we're selecting materials that the cells are familiar with. For example, one of our inks that we routinely use is a fibrinogen-based ink. Fibrinogen, for those of you that are biologists, may remember that it's something that's found circulating in your body. So when you cut yourself, fibrinogen interacts with thrombin and it forms fibrin, which is a clot. Which is, what's nice about using fibrin as a print material is that in the body, whenever cells see fibrin, they know that something has happened because there's a wound or whatever, and so the cells get to work. They decide, hey, you know, there's a wound here, now there's this fibrin clot that's temporarily blocking the blood from spilling out, now we've got to fix this and make this our own. So it's a material that gets cells, you know, moving and reconstructing. And that's exactly what we want to do. We want to use a material that gets the cells to self-assemble. So in this case, we have two types of cells, blue and yellow cells, that initially are not in any kind of orientation. They're just printed dispersed. And then you see the results. On your left, you have endothelial cells that came from umbilical cords, and then on the right, you have endothelial cells that came from the brain. Now, what's pretty obvious is that they're both different. And this isn't necessarily a problem, right? Because I said that the vasculature is very different. So it's a good thing that the brain cells look very different than the endothelial cells that are from the umbilical cord. So this is a good type of ink because it lets the cells do them, lets them be themselves. Now, if we want to do non-planar, meaning we don't want to be in just one plane, we have a type of support bath ink. One of the challenges with printing materials that are found in the body is that they're pretty soft, right? Like if you think about yourself, you're pretty soft in most places, right? So the thing is, if you're going to print with material such as collagen or fibrinogen, it tends to be kind of soft. So if you want to do layer by layer, it becomes kind of challenging. And so a ink like this is really useful because you can print into it in 3D. So what it is, it's a support bath. I like to tell people, like, think about being in a ball pit, right? Like when you jump into a ball pit, you don't sink to the bottom because you've got sort of like all these different ball particles that hold you up. So we kind of use that type of idea for this support bath. So we can print into it and print up in 3D because it's sort of a support system that holds it up. Going back to the ball pit analogy, imagine now that you're in the ball pit and that then we fuse all of those balls together. And now you've got a surrounding support system that holds still the really soft system in the inside. And so that's what the support bath non-planar 3D printing is. Okay. So I said the 3D printing is great because it actually provides structure. So let's talk about the inks that we use to provide structure. We, in particularly, are interested in the vasculature because, as I mentioned before, that's really important if you're trying to build up a tissue. So the way that we provide structure or channels is a couple different approaches. One is using a coaxial coaxial nozzle to print tubes. And coaxial nozzle is just a fancy word for saying we have a needle within another needle. So on the outside needle, we'll introduce a material, and then on the inside, we'll introduce a cross-linking solution so that when it's being extruded, the inside is cross-linking with the other material on the outside, and you end up with a hollow tube. So that's what you see on the picture up there on the left. We can then take that hollow tube and introduce cells on the inside, and so in this case we introduce endothelial cells, that's what's the green, and now you have essentially a blood vessel because you've got the endothelial cells that are lining, and then on the outside that we printed happen to be fibroblasts, so a support cell on the outside. This is great, but we're kind of limited to just having straight tubes. So if we want to make much more complex geometries, which is definitely what you find in your body, it's not just a bunch of straight tubes, then something like a fugitive ink works. And fugitive ink sounds kind of, like a detective or something, but essentially what it means is it's an ink that doesn't stick around for long. You just use it momentarily. So in this case, we use the fugitive ink to sort of lay down the pattern that we want, and then, you know what, I'm going to use a pointer if I remember where I've left it. Oh, there it is, okay. So we can print out the geometry that we want, and then we can print around it the other structure. And this is where the fugitive ink comes into play, because we then melt it away. And this particular ink is interesting because it melts at cold temperatures, which sounds a little bit weird, right? But it works great for us because we can print something at room temperature like this, print everything else that we want around it, and then stick it in the refrigerator and it empties out. So here's a quick video showing that. So right now, what you don't see is that there's a pattern here, and we're pushing, in this case it's dye, but normally it would be media, we're pushing it through so that it displaces the fugitive ink, and what's left now is a hollow channel. This hollow channel can then be seated with endothelial cells, and now you again have some kind of a blood vessel that isn't limited to just being circular. Okay. So the other big elephant in the room is, this is all great and pretty, but this bowl of spaghetti-looking vasculature, is that really what our body looks like? No, of course not. What our body has in terms of vasculature is very much a hierarchy and a pattern. You have really large arteries and veins that become arterioles and venules that become capillaries. The way that our body designs that is based on pressure profiles and concentration gradients. So if you want to have this vasculature look like this, what you need is to be able to create a dynamic environment. And what I mean by dynamic environment, I mean one where stuff is moving around. And that's, pretty different from traditional biology, where you think about, seating something in a plate and then putting media on top. Those are very static systems. And that's not at all what the body is like. Inside of our body, there's stuff moving through our blood vessels, and in between our blood vessels, we have what's called interstitial flow. So the point is, there's always movement going on inside your body, and so that movement is what gives the cells the cues to arrange in the positions that they're supposed to. Changing things a little bit is, we don't just print things that are alive, we also print with inert materials that are not alive, and in this case, we're printing with PDMS. The reason we're printing with PDMS is because we want to sort of design our own petri dishes. I told you that the traditional bio of like putting something in a plate and putting media on it isn't going to cut it for our dynamic vasculature. So we almost had to reinvent what those petri dishes are going to look like, because we now want to be able to connect it up with fluidics. And so we can print with PDMS, and we can do almost any kind of geometry. On the top here you see one where we have two different channels, and on the one in the bottom we have an option where we're only going to have one inlet and one outlet. So we can have something come in, branch out, and then come out the other way, or we can do sort of like that video was showing, two different ones where we can play around with the concentration gradients. So essentially this gives us a lot of flexibility with how we're designing our tissues, because we can essentially print the petri dish that we're putting them in, decide how many inlets and outlets we're going to have. Ooh, don't go to bed. Hold on. Sorry, look away, look away. Okay. Oh, no. Sorry, I get to see my incredibly messy desktop. Okay, sorry for that technicality difficulty. Let me cue us up. Oh, hang on. Okay. All right. Okay. Okay, so I was, let's see, where was I at? So yeah, I mean, basically the point is, it's pretty cool because there's a lot of different controls that we can do. We can print everything. So now moving on to where is this all happening? So we have a bioprinting facility. This is the lab. I'm sure Sierra's probably thinking, what lab is that? Because it has definitely grown, and there's definitely a lot more people, and sometimes it can get a little messy. But it's a pretty cool lab. It kind of has both biology stuff as well as engineering things. So we have everything you'd expect to find in a biology lab: incubator, biosafety cabinets, and then you've got a lot of the cool engineering toys like a confocal microscope, a rheometer, which is just a tool that measures how viscous something is. What other things do we have in there? Like different microscopes. Oh, the printer. So here's where we put the little printer, the sort of MakerBot. We've upgraded it now. But then this beast over here is the printer that we mostly print on. And it's a fancy Aerotech system, but essentially it's like the MakerBot in the sense that it controls X, Y, Z. we had to build a bubble around it because we are printing with things that are alive, so it's important to be able to keep things sterile and also to protect ourselves because we are printing with things that are from human sources. And so the inside bubble offers what we call product protection, meaning that it's a positive airflow, so it's pushing things away. But you don't want that stuff to be pushed away and onto you, so we then build another bubble around it, which is the negative air that sucks stuff that's being pushed out. So even trying to get this printer up and going, we had to do a little bit of engineering to get it to work the way we want to do it. Okay. So now we move on to the cool part, which is like, how in the world are you doing all of this? What should be kind of apparent by now is that it's kind of a really multidisciplinary type problem, right? Because I've been talking a lot about biology, but a lot of engineering. And so you end up having to be working with things that are, again, biology, but also mechanical testing and material design, a little bit of computer modeling. And so it's sort of a feedback mechanism in the sense that we're building biology the way that we think biology exists, right? And a lot of that is gleaned from our static cultures as well as from animal studies and from, in vivo observations of human physiology. But we're still kind of limited, right, in how much we really understand biology. So I'm trying to put together something that I kind of think I understand how it works. And what's cool about this approach is that as I'm putting all of this together and manipulating it, I'm going to find out some really cool things about biology that we just haven't even discovered because we've only been doing things in a dish or only been doing things on animals which are not directly don't make a direct correlation to human. So I kind of see this as an iterative process because I'm building biology to understand biology, but I'm going to find out different things about biology that are then going to influence how I build the structures in the first place. So it's kind of this really cool, crazy feedback mechanism. Now this is where I'm going to turn things over to Javier because this is the part that gets kind of cool. Like this is the day-to-day stuff. How in the world do I balance it between the engineering and the biology? As I mentioned before, the game is to keep cells happy throughout the entire process, make sure these guys are smiling all the way through. Because what we're printing them in affects how they're going to experience this process, right? Because think about being on a water slide, right? If you're going down a water slide in water, super fun. If I send you down that water slide in honey, maybe it's still a little fun. If I send you down in cement, not as fun. So it's kind of the same thing. So when we're looking at these materials, we have to see, okay, what kind of, slide material are we sending them down? And are they staying happy? And then, of course, as I mentioned, after you print it, you have to make sure that whatever you printed them with is something that they're going to enjoy interacting with, because then that becomes their home. And so it's this sort of crazy biology versus engineering, well, maybe not versus, I would say working with engineering. And so Javier is, as was the introduction indicated, he's an engineering technician in my group, and he has been tasked with the assignment of developing a bio-ink and making it very specific to the brain. As I mentioned before, we do different types of inks and we're targeting different types of organs. And so I gave him the challenge of, take one of our inks and make it into an environment that brain cells are going to want to grow into. And so I'm going to have him tell you a little bit about what kind of process he uses to try to get there, and also maybe share with you some of the data of the bio-ink that we've been working with. So there you go.
Hello, testing? Oh, God. Okay, so before I talk about bioprinting, which I'm actually really excited to talk about, I do want to say that a lot of the work that I'm going to be telling you about is stuff that I started working on as an intern, kind of as like a side summer project, and it kind of blossomed from there. And so now that I'm like a full-time employee, it's still kind of my pet project, and we've tried getting it ready for, handing it off to other people to work on, and I'm struggling to hold this microphone. But it wasn't my first project. So I came in with a lot of biology skills, and one of the first things that Monica threw me into was engineering and working on fluidics and that sort of thing. And I'm really grateful that she did that, because it ultimately changed the way that I thought about things. It helped me develop the way I approach problems in a more methodical manner. And that kind of gave me the key to really go forward and figure out the problems that we were having. So I have a slide here that talks about the scientific method and the engineering method. Most people are probably familiar with the scientific method if you ever did like a science fair project. And if you ever did a intro to engineering class, you probably were introduced to the engineering method. Since bioprinting is interdisciplinary, you need to have two ways of thinking. You need to be able to throw on your science hat, but you also have to be able to switch it out and put the engineering hat on. And so that's where these two things come into play. One's not necessarily better than the other. I think that the key is knowing when to switch the hat. But the key to both really is iteration, right? Always working and improving on what you got. Oh. So this is to highlight the fact that one can inform the other, right? Yeah. Oop. And yeah, iteration is key. So what's a bioink, since that's what we're developing? And since we're developing, we're using the engineering design process, right? So what is it? Let's do some background. So a bioink is essentially something that has cells and is a structural material. So you need those two components. And specifically, I'm trying to develop a bioink that will give these cells the kind of signals that they would normally have in the body, so they need to behave like they normally do. But at the same time, we have to recognize that what works for one cell does not work for the other, right? So these are both endothelial cells that Monica mentioned, and they're essentially cultured the same way. They have the same material, but the problem is they don't really look that great on the right? The brain cells, let's see, the brain endothelial cells, they're not really forming the networks that we want to see, like these guys. Sure, it's not structured very well, but I mean, look at there's just not a whole lot of branching. So my task was to find a material that would get them to, look like they should. So what should it look like? So since I'm developing a neurovascular bioink, I should, talk about what a neurovascular is. So the neurovascular unit essentially is the blood vessels in your brain. But specifically, it's a little bit more than that. It's multicellular, so you have a neural component and you have a vascular component. And it's the interaction between the two that really makes it exciting in what it is. So that's the neurovascular unit. So what are the key players? At the bare minimum, you really need the endothelial cells, right? Because they make the blood vessels. But they have a lot of buddies. Our focus should be the astrocytes, since they're the ones that are more, physically entwined, I should say. So now that we have an idea of what the neurovascular unit is and what it does, it helps connect the blood of the system along with the brain cells. We should probably think about how the blood vessels will normally develop. So Monica mentioned earlier that one of the key approaches is taking cells in a gel and letting them do their thing. And you can get different structures from that. Blood vessels typically form in one of two ways. They form two different ways in the body. So you have vasculogenesis, where you have cells that are just kind of floating around there, forming structures, and ultimately that forms the blood vessels. So that's vasculogenesis. And that's what the HUVECs do really well, the other ones that were making all those branches. But then there's another way that actually happens a lot more in the adult body, and that's angiogenesis. And that's where you have pre-existing vessels, and the new vessels will come from those by sprouting, kind of like the branches on a tree. And if you look into the literature and you read papers and books, you'll find actually that brain vasculature forms through angiogenesis. So our normal approach of taking cells in a gel might not apply very well. So taking that into consideration and getting our bio talk out of the way, let's think about our mechanical needs, right? So what are the rheological and mechanical properties that we need in a bioink? What gives it its structure? And so that's usually termed as printability. So what makes an ink printable? That depends on the method being used. But since we're doing extrusion, specifically we're looking for something that's shear thinning. So that property is where it flows when you apply stress to it. So if you think of toothpaste, that's usually a really good example, right? You have to push it out the tube, but if you hold the tube upside down, it doesn't spill out. Rapid gelation, so it needs to turn solid, right? So you have the cells floating around. Once it's printed, you want them to stay where they are, essentially. You don't want them to sink to the bottom during the print. You don't want them to sink to the bottom after the print either. So it needs to go from liquid to solid. And then after you do that, does it hold its shape? Does it hold its shape? So if you print something, is it still going to look the same after you immerse it in liquid or some other substrate? And so how do we determine all these properties? Well, we use the study of rheology, and that tells you a lot about how properties will flow or how materials will flow and if they'll deform. So that tells you, that can inform you if the processing is affecting the structure and how that structure will affect its properties. It's our main tool for that purpose. So using rheology, you can also find different things, right? So how long does it take to gel and how stiff does it get? Because those are also considerations that we need to look into. So this graph on the left shows you that if you increase the concentration of fibrinogen, it takes longer to gel, which is not a good thing, right? Because now you have to balance that with knowing that the more concentrated it is, the stiffer it'll get. And if you want something that's really stiff, the brain vasculature environment actually is like 1,000 pascals. I don't know if you guys know what that is. I'm not sure if I do. It's pretty soft, right? For those of you taking materials engineering, like 1 kilopascal is pretty low. But if you have it, if you're trying to achieve that with just one material, you can't do that, especially with fibrinogen. And so taking all these things into consideration, this is kind of what data looks like. You can take this information, make a chart, and you have a pretty good understanding of what your materials look like and how you can use them. So we have these biological constraints, and then we have these mechanical constraints. So what do you tackle first? Do you tackle, can you do multiple things at the same time? So this is like balancing, it's a delicate balancing act. Let's leave it at that. Why does it have that sign? That's a weird emoji. Anyway, so when we're looking at the biological constraints, let's think about, so we're concerned, are the materials that we're putting the cells in toxic? Are they poisonous to the cell? Is it affecting them negatively? The next question is it biocompatible? What does that mean? Why is that different than toxicity? Well, you have toxicity, and then you have whether something is bioinert, right? Is it giving them proper cues, or is it not affecting them at all? So that's another thing you need to think about. And then selfishly, we're really interested in, does it encourage them to form blood vessels? Degradation of the material is important because if they can't break it down, they can't remodel it, right? So the cells are really notorious for being able to do that. If you put them in something that they can't interact with, they'll be stuck. And if you look in the microscope, they're all balled up. Whoops, wrong way. But if they can degrade it too well, then they won't be able to build anything in it. It'll be like trying to put a house in quicksand. It ain't gonna work. So I'm just going to click through these real quick. So mechanical constraints. As I mentioned, you can use rheology to find out what the properties are and what forces you need to kind of get the material to flow. Well, if you need a really high amount of shear or just force, you're going to kill your cells because they're not going to like that. That's like pushing someone down the slide. Cross-linkable. So what strategies can you use to take something that's liquid and turn it into a solid? So Monica talked about using enzymes or chemicals to make that change, cross-linkable, a cross-linker essentially. And in the end, what's the final stiffness? Is it going to match tissue? Does it matter? Sometimes it does. And kind of like stability, does it hold its shape after the print? Those two. Oh, yeah. So what's the secret to balancing the biological with the mechanical? Not that sauce. The secret to our flavor of bioprinting is keeping it real. That is, we don't use synthetic materials. We use materials that are biological in nature. So specifically, we use gelatin and fibrinogen. Fibrinogen will help us get the biological signals that we want, and the gelatin, which is really similar to collagen, gives us a lot of the mechanical stiffness that we need, and in general, it helps you. It helps having that combined material. All right. So now that we have some basis for what our materials might be, we need to think about the cells and how do we get these ingredients just right. The neurovascular unit is multicellular, but what cells are important and how do we test for this? So we culture these in what we call spheroids. Clearly, they're not very spherical. But this allows us a couple of things. So since the brain vasculature develops through angiogenic processes, we can kind of use this as like a point source. So if they sprout, then we know that they're doing angiogenesis. And so we can look at these once they're embedded in a material, and if they're reaching out in some way, then we have pretty good indication that they're going to interact with something. This also allows us to, well, it actually allows the cells to assemble the way that they would naturally. So they're interacting with their buddies in close physical contact, and they can rearrange themselves as they see fit. So even though we have our materials and our cells, we find that the source of the materials can have an effect on what they look like. So Monica mentioned that we use fibrin, which is derived from blood clots. So if you have fibrin that you get from a cow, or a mammalian-based fibrin, the cells don't seem to like it as much, which is interesting because this is kind of the basis for not only our process, but several other tissue engineering methods. And so if you're trying to put brain vasculature into this material, it doesn't seem like the cells like it very much. And you can determine that by, one, this really sad little sprout that's kind of going away. It's like a seed coming out and then dying. And then on the right, you got this sprout that looks like it's about to give you a high five. And this is only 3 days in culture. We usually let these experiments go for like up to a week. I've sometimes let them go a little longer, just for fun. But clearly, even after a week, there's not a whole lot of change. These cells just don't like the mammalian-based fibrin. And they like salmon. But who can blame them? So you saw that sprout was kind of chunky, right? So the sprouting is a good direction, but it tells us something's still missing. And so if we add their buddies into the equation, if we throw some astrocytes into it, we start getting more defined sprouts and a lot more sprouts. And that's really cool. That's exactly what we want, at least as a starting point. If you look at these cells, you see a lot of sprouting, but it doesn't really go very far. And it just got this weird shape to it. But this is only day 3, so let's see what happens over time. So, holy moly. All those sprouts that we saw, if you remember in the other picture, you see the color in the center, and you can't really see who's who in the branches. But this confocal image that I think Sierra may have taken, actually. Yeah, she recognizes it. It probably took her 3 hours, I would say. So that whole spheroid is about 1 millimeter. So you can actually see these if you hold up the gel to the light. So you can see this mass of cells. This is pretty big too, but as you can see, there's not a whole lot of things happening. The astrocytes seem to be okay. In fact, they're probably a little brighter, but overall, the structure that we're looking for is something like this. So now we kind of have to put our science hat on and say, why do they like the salmon more than the bovine? Is it a mechanical thing? Is it a chemical thing? And how do we test for this? So the easiest way to go about this is obviously to match the stiffness, assuming that the stiffness isn't the same. So for all this time, we were going back and forth in the lab. Is one more stiff than the other? What is it? Is it a chemical thing? So we decided to kind of just put it all on the table and do rheology. And so all 5 mg per mil. When we look at the differences between salmon and bovine, huge difference in terms of stiffness. It's still not 1 kilopascal, so it's not exactly tissue mimetic, but it's still a strong indication that maybe they like something that's more stiff. So the reason why I put all of these different tests that we did was to see which ones we can match up really well. So most of our processes actually use 10 mg per mil. So we were trying to find something that's close enough to that and see what would happen. So we decided to go with 3 mg per mil. And as it turns out, making the gel softer didn't really have a negative effect. They still did a lot of branching, and they did it in a faster time scan. So reducing the stiffness, the gel concentration didn't have a negative effect. So it's not just mechanical. So clearly it's something biological. So we learned a lot, right? So we know the material is important. We know what concentrations that they need and what we can use, and we know what cell conditions they need. At bare minimum, we need the astrocytes and the endothelial cells. So the next step, obviously, in the process is to take all this information, put it back into our process, and optimize. And so Project Maki is born.
Okay. Thank you, Javier. So he kind of gave you guys a little bit of the behind the scenes, like sort of what we actually struggle with, right? Because I gave you the hype part, right? Telling you, oh, it's great, this is what we're doing. And then he really gave you an example of, you know, what that all means. How do you break that down? How do you even begin? So I'm going to continue on and do the hype piece, meaning telling you about the exciting things and getting to the final question of, okay, so why, again, are you bioprinting? Are you really trying to make organs? Does the government really want to print organs? The answer is no. That's not what we're in the business of. What we really see the value in these bioprinted tissues is that we're essentially creating miniature versions of organs. And the reason we're doing this is because we want to understand human physiology. And the reason, of course, that we want to understand human physiology is because at the lab we have a mission space that focuses on biosecurity. We want to understand how different toxins and chemicals affect human physiology. Usually the way the drug industry decides how something affects a person is that they try it out on an animal first, and then onto a person, right? They move into clinical trials. Well, the problem with things like developing medical countermeasures is that you definitely don't want to have a trial group, right? Like you want to know that something works or doesn't when you need it. And so what we really are missing is having a way of testing something on humans but not really humans, right? So this is where bioprinting can particularly play a role. The other thing too is, for those of you that are familiar with Lawrence Livermore National Laboratory, is that we have a lot of really fast and powerful computers. And so the idea is, what if we were to take some of those powerful computing power and match it up with experiments? Meaning, what if we didn't have to do every single experiment to find out how something is going to work? What if we could model some of that biology and find what is the most efficient or maybe what are the experiments that are going to matter more, that are going to make a difference. And so that's kind of where we fall into the model and make iteration of going back and forth. But then in order to actually understand what's happening, you need to be able to make measurements. And so that's kind of where we see bioprinting fitting, is that we want to be able to help build up our computational models with these experimental models. So I'm going to tell you a little bit about the 2 bioprinting projects that we have. One of the bioprinting projects that we have, which is the one that Javier works, well, actually Javier works on all of them, but the neuro ink that he was talking about was for this project. And in this project, we asked the provocative question, what if we could predict where cancer is going to metastasize? Cancer is horrible, certainly when it moves into the metastatic stage, meaning when it spreads to vital organs like the brain. That's pretty much when it's game over. But what if we could tell where that was going to happen? Especially in the brain, because when stuff metastasizes into the brain, it's really hard to track. Normally with a cancer, the way that they find that you have a big tumor is that they'll introduce a dye into you, and when they see that the vessels are leaky, they say, that's where the tumor is. The problem with glioblastoma tumors is that they hang out very close to the blood-brain barrier, the vasculature that Javier was talking about, and they essentially don't create their own vasculature. They do when they get really, really big, and the problem is that if we don't detect it early enough, then it becomes harder to treat. So that's a provocative question that we were asking, and the reason we asked it is because it turns out that people don't really know why certain cancers like to go to the brain. The biologists have definitely looked at what's called the soil and seed method, which is they're trying to figure out what is it about the environment, what is the stuff that they're secreting that is calling the tumors to that area. Then there's another theory that says, well, if you look at it, it sometimes tends to be in very particular areas, like where the vasculature branches. So maybe there's something about the architecture that is determining where it's getting stuck. As I mentioned, it's super easy to look at what is being secreted because it's easy to look at that in a plate. What's a lot harder to do is to look at the architecture and the role that the dynamic movement is playing in determining where that cancer is going to get stuck. And so, simple static culture isn't going to do it because we're talking about a process where stuff is moving. People use animal models, but the problem with injecting cancer into the animal is that it's going to eventually pass away because that cancer is going to go to vital organs. And so it's problematic because if you want to observe that metastatic process in a living organism, you could potentially take them out before you're even done with your experiment. People have started to use microfluidic models because that's one way that we can introduce flow, but the problem with microfluidic models is that they're very constrained. You dictate the geometry and that's pretty much it. And so what's good about us is that we can dictate the architecture and also keep it biologically relevant. So the way that we see the flow processes is that we're going to have a computational model that's going to model what we see in the printed tissue. And these models are pretty complicated. Like, they take into account that you have red blood vessels flowing, they take into account that your endothelium has some kind of elasticity, stickiness, and whatnot. So it definitely requires high performance computing. And we want to build that up with the experimental model. Now, I don't know if any of you have a background in computers, or in data or computer engineering, but one of the big problems is that if you put junk into your model, you're going to get junk out. And biology tends to be incredibly messy. One of the cool things about bioprinting is that I can simplify it to something very, very simple and validate that, and then increase the complexity, right? Because I can start off with a very simple geometry, make sure that the model works, and then increase it up and build it up. And so this is what the process that we're using to be able to create a model that will eventually help us predict where to look for the brain mets. And the video is not showing very well, but here's a picture of a branch channel that we've printed, and you might see like movement, but what you're seeing is 1 micron beads that are flowing through the vasculature channel. The point is that it's kind of cool because you can see that this channel is responding the way you would expect it to respond in the body, meaning it's got some kind of a compliance. Like, this is clearly a better model than just printing something that is not biological. And we can see the cells on there, so Javier was talking about making sure that the ink is one that encourages them to remodel. So we can see the cells, and we do see that they actually respond to flow, meaning they line up in the direction of flow. And here are the endothelial cells that are starting to branch into the surrounding area. So that's where we're at right now with the Brain Mets project. I forgot to mention that Amanda Randles is the collaborator that I work with at Duke University, and she's got postdocs and grad students that are helping us out with the computational model. Okay, so the second project that we have is a 3D bioprinted tumor. So for this, what we're thinking is you take a tumor from a person, which is, if you think about people that have cancer, they go in for a biopsy. Well, what if you then grew that tumor in a bioprinted environment, and now you have a little test version of you specifically. So now we're talking about more precision medicine. And again, like I said, we love computers, and so we are also trying to model this. And so we have an in silico effort as well as an in vitro effort, meaning in silico is we've got people that are helping us understand what we're observing in the biology realm of things and seeing if we can create models that can help us predict. So for example, with this tumor, in addition to bioprinting the tumor and doing some, dynamic biological assays, we're also going to integrate them with some of our sensors that we have at the lab. I think before someone has come to talk about some of these implantable electrodes that we use to put into people's brains, where we're using that same technology to probe into the bioprinted tumor. The idea being that we can get some kinds of measurements from there. For example, what's the oxygen tension, pH, stiffness, how are all those things changing over time? Feed that back into a computational model and create something that's able to predict, for example, the likelihood of a drug working specifically for the patient itself. So where are we with that project? So with that project, we have, hang on. Oh, sorry, I did not realize it was animated. We have bioprinted these tumor spheroids. Again, the pictures don't show up really clearly, but here you see the little diamond-shaped reactor that we have, and that red line is where we have the vasculature. What we found interesting was that the tumors that were right close to the vasculature were kind of these big balls, whereas the ones that were furthest away, the tumors were very, what we call invasive and had a metastatic phenotype, meaning they were very spidery looking. They had all these little branches. And that's pretty cool because you can have exact same cells with the exact same material, but depending how far away they are from the nutrient source, you're going to get very different phenotypes. So that, again, goes back to the point that you can't leave out the vasculature piece because that plays a huge role in even the way the cancer progresses. So that's what we have on the make side of things. And on the things of measure, what we want to do, of course, is get very, very, very close to the human, which is the top right? Biology gives us this, 2D culture. What we're hoping is that the bioprinted tumor is closer to the human. Like, we're never going to get to the human, right? I'm never going to bioprint an entire human. And that's where the computational model comes in, right? I'm going to get pretty close to the human and then use computational models to fill in that gap. So between bioprinted tumor and the human that I'm trying to replicate, that's where the computational model comes into place. And so one of the things that we're very cognizant of is we want to make sure that what we're bioprinting actually looks like what it's supposed to be in human. And so we have a whole bunch of genomic data that we're, one more thing here, there we go, a whole bunch of genomic data that we're looking at to see, are these cells still looking like what you'd find in the tumor? And so we are also comparing that to the 2D cultures as well as to the animal models, just to better understand, like, how do all these different tools that biology uses, how do those in the end approximate human physiology? So here's a close-up of one of our bioprinted tumors. You see the channel in the middle, and then you see the tumor spheroids, which are circled in green. And here's a close-up of our tumors. What's really interesting is you see that little halo? Like, that's not something I photoshopped in. That's actually a remodeling of the environment, which is pretty cool because tumors normally remodel their environment. Tumors make the environment very stiff. So if you've ever, people that end up getting cancer, one of the ways that people know that it's cancer is that the doctor feels for the tumor. And they can tell based on how it feels, whether we're talking about a real tumor or whether it's just something like like a lipid or something. And so this is interesting because we have evidence that these tumors are actually changing that ECM that we printed them into. And so that makes us excited because that means that the tumors are behaving the way we expect them to behave in the body. So, like I said, everybody keeps thinking, we're going to be able to print organs on demand, and sure, I think that that's definitely the end goal for 3D bioprinting, but if you ask me, I think it's the path that gets us there where we're going to see a lot of really cool discovery. Today, most people that are bioprinting are doing these small-scale tissues like we are for drug development, and in our particular case, we're more focused on the vasculature piece of it. But I think in the future, we might actually start to see actual simple tissues that might be paired up with cool technology. So, I mean, who says that we have to print the liver exactly the way the liver is? What if we can print like for the older people in the audience, RoboCop, if you ever remember RoboCop, where like they put together a person, right? And it wasn't just a person, again, the person had like superpowers. So I think that the cool thing about 3D bioprinting is that maybe we can replicate some function of organs and maybe make them better. Maybe we can do a pancreatic patch or something that secretes insulin, but maybe also provides information about the changes in insulin, because we can instrument them with some of our technology that we have now. So I actually think that's pretty close in the future for happening. One of the other things too that we're, especially from the lab's perspective, that would be interesting is, I talked about having these models to test drugs, but what if we could use these models also as human canaries, right? Like the canary idea is that people in the miners take these canaries to see if there's something toxic, if the canary dies, and they know, like, we better get out of here. So what if we could use these tissues also to help us understand if there's something toxic in the environment? So that's really where I think we're going to see some really cool technology emerge, and I think we'll be able to do more than just replicate biology the way that we understand it. And so that pretty much covers it. I see we're already at 7 o'clock, and I want to make sure that we have time for questions. So thank you so much for being such a captivating audience.
Interesting things. Any questions? This is your chance. Okay.
Hi, thank you for your awesome presentation. I was really interested in it. But more importantly, when you guys were talking about neurovascular bioink and how it's really important to generate the tissues to create vascularization. When you're talking about the brain and how all of the epithelial cells connect with astrocytes, is that important for vascularization because of the astrocytes are kind of like the pipeline that kind of bring in all the nutrients and the oxygen?
Yeah, actually, that's a really good point you bring up. The astrocytes are incredibly crucial to the blood flow. Like, they can make the blood vessels become more leaky, which calls more blood flow to the brain. So yeah, the astrocytes are super crucial for even dictating, for example, how much blood flow your brain is getting. They can start to secrete chemicals that maybe make the blood vessel a little bit leakier and causes the redirection of flow up to the brain. So yeah, that's exactly why, as Javier was saying, that the two minimal ones that we were going to start off with was endothelial cells and astrocytes. Cool. Yeah.
We have one over here. Can you pass down the...
Hello? Okay, cool. You said that you wanted to use these 3D printing in order to potentially see how medications or drugs would affect the body. When it comes to those models, would they be going through the same process in which, like, there's different, you know how there's different administrations of drugs, and if you go through intravenously or you go through it through the GI tract, it causes the first pass effect. Would those 3D models and printings also be able to account for the body's reactions and changing with those drugs?
That's a really good question. You bring up two really important points. One is, if I'm talking about using these to replace animal testing, there's an administration piece of it, like, well, how would the FDA, for example, feel about that? And then there's the other point of, like, which of these organs are you actually going to create? Because, like you said, some of the stuff is taken up in the GI, in different parts of the GI. So there's lots of people that are working on different parts of the organs because they want to see, for example, this drug is taken up in the GI, but it may have a cardiotoxic effect. And how do you also then pair the cardiac system with the GI system? So on a project I didn't talk about was the Organ on a Chip project that both Javier and I are a part of. And in that one, we create different organ systems. Again, it's related to the brain. So we do have a heart organ, or not a heart organ, we have a heart on a chip device because we are interested in seeing, okay, if we're going to give certain drugs to the brain, which of those are going to also have a toxic effect. So yes, when we're trying to put all the pieces together, we need to be able to have almost all the other missing organs as well, because even if it's going to be administered in one area, it may have an effect on other parts. Now the other part is sort of like, how realistic is this in terms of actually being something that drug companies can use? The thing is with animal studies, they're not even that great either. When you think about how long it takes a drug to develop, it takes anywhere from 10 to 15 years to get a drug through all of that process. And of the only 10% of the drugs that actually make it into animal studies end up succeeding. Meaning you've got 90% failure. What's that?
$40 million per human testing.
Right, exactly. And so like the drug companies are super excited about this because this potentially is could potentially be more like human physiology and will definitely be cheaper than animal studies. And so that's, I get asked that question and I think, well, what we have now isn't that great either. And what's interesting is we've actually partnered up with a drug company called GSK, and we have a project that's called the Atom Project. And working with that company, we've taken on the challenge of seeing, can we get that 20-year process down to a year? And one of the things that we're doing is we are using computational models to help us understand that. And one of the things that we're doing is we're taking a look at all of their assays and trying to look at their failed drugs and see, could you have predicted success or failure? Which of these assays would have been actually beneficial and which one of these are just a waste of your time? And so we have a really big effort with them to try to reduce it down to 1 year. And a key part is using what we call these complex in vitro models to better understand human physiology. I think, though, we have a really nice little test case that we're doing, and we're going to start initially with cancer. And we are looking, through the lines of like, how do we get the FDA to allow us to sort of streamline this, right? That we're not going to do all of the animal studies that they require to do before we can actually test into patients. So, yeah, that's a really important piece of all of this sort of drug development platform. So, really good questions. Thanks.
Any other questions?
This is for the fourth dimension. How long can you keep the cells alive?
Let's see. So for the flow ones, we've kept them 3 weeks?
I think the
4?
highest so far is 26 days.
26 days for under flow. Under static cultures, we've gone up to 45 days, and basically it's because we want to see what happens, and so we off them before they have a chance to fail on their own, just because we're anxious to see what's going on. With the tumor project, we're starting to implement these sensors so that we don't have to try to finish the experiment to see what's happening. So to be able to tell what's happening and how they've grown, we do staining, but we have to fix those tissues. So a lot of the assays that we're doing are terminal. And so we want to integrate more of the sensors that we have so that we can keep them for longer and see what's actually happening without having to take it apart and see what's happening.
Any other questions? There's one over there.
First of all, thank you very much for your wonderful talk. I wanted to ask Javier and also maybe Sierra, because you're the most close to the people in this room in terms of your academic and work experience pathway, what's it like to transition from college student to intern to now working full-time? And I'd really love to hear from both of you about where you are in your path, and maybe also to talk a little bit about failure. Because I'm guessing you had to do some of these experiments more than once.
Oh, yeah. Yeah. Let's see. There was a lot of questions. I don't know if I was able to remember them all, but so going from school and classes to transitioning to essentially full-time work as an intern was a challenge in terms of like being able to, when you're a student, you can kind of like disengage and you focus really intensely for maybe an hour and then or 2 hours, and then afterwards you go and listen to music or hang out with your friends. But when you're actually working, you kind of have to stay there and you have to actually keep making progress on things. So you're reading papers or you're having conversations about how something failed, right? What happened? What went wrong? What didn't we think about? And I saw this paper and they did this. Let's try this. So then you move on to the next thing. You start prepping experiments. Since we're working with a lot of living materials, you'll start an experiment by like seeding the cells, and you have to take care of it over the course of maybe a couple weeks before you can actually use them. And so that time scale is something that you're probably not used to when you're a student, right? When you need to actually do your thing, you do it. Whereas for this kind of work, you kind of have to prep weeks in advance sometimes. Don't know if that answered all the questions. Hey, Sierra's here.
Yeah. What exactly was the question again?
Well, I think so they were asking about sort of like the transition and then, failures and stuff. But I think one of the things I want to mention is that both Sierra and Javier have been working and going to school. So Javier took the semester off now, but I'm curious as how you do a balance, because at least in my trajectory, I did school and then worked. But both of you have actually balanced both school and work.
Yeah, so I'm actually a full-time student here still. I'm in the mechanical engineering technology program, and I'm graduating at the end of this semester. And honestly, if I had like a full-time job or even a part-time job that wasn't willing to like work with me like the lab is, there's no way I would have been able to do it. Like right now, as far as scheduling goes, I'm at school the majority of the time, and I work like 3 hours on Tuesday and then like 5 hours on Friday and a full day on Thursday, just because that's how the schedule worked out. And if I didn't have the flexibility of that, or if I had to work weekends or whatever, I really wouldn't have the time to balance all of it. But because I was lucky enough to be in a job where they do want to work with you on scheduling, it's much easier to balance it. And honestly, because I have a job in my field, it's nice to kind of get that, I don't know if reassurance is the right word, but like backup. Like if I'm learning something in chemistry or in physics, and then I go to work and I'm actually doing something that is applying those same principles, it really helps, and I feel like that helps balance the work and school kind of system.
Congratulations on graduating.
Thank you. I haven't done it yet. I still have to pass physics.
Are there any other questions from the audience?
Hi. I don't need a microphone. I guess I'm just wondering, because, like, as a scientist, like, how do you feel about, like, all of, like, how do you, like, not, like, how do you overcome the obstacles from, like, other people who think, oh my gosh, they're, making clones at the national lab or something like that. I mean, I'm a business student, so it's my first semester taking botany, and I'm like, open my eyes, and I love it. It's my favorite class I've ever taken in the whole trajectory that I've been here in Las Positas. And I'm just wondering, I'm like, well, now I want an internship at the national lab, because it just seems like it's like a right fit for me, and I'm sure there's a business aspect of it. But, dealing with like the FDA and activists and people who think that you're doing something wrong there, like how do you do that, and how does that how do you overcome the obstacles so that way you can focus on the actual work that you're trying to do?
Well, one of the good things is that we do a lot of community outreach to inform the community of the kind of work that we do, because a lot of people do look at us and think, oh, you're just building bombs, because that's traditionally what the lab originally started on. And so it's kind of funny because every now and then we'll have protests that will actually like even shut down part of where we can come into because they'll, put themselves on the ground and whatnot. So I think one of the really important things is doing community outreach like this, informing people of that we do more than, well, we don't make bombs anymore, but we do more than just what traditionally the lab was designed for. We do a lot in the biology space and the engineering space. And even biology has had a really interesting path. Initially, it started off with we wanted to see what were the effects of these nuclear bombs on human physiology, and so that's kind of how the bio program started. And then we were interested in like, well, how do we detect what's in the air that could potentially hurt humans? And now we're at the point where we want to understand human physiology, where we're not so into, I mean, we still do a lot of the detection stuff, but we're interested essentially on what the effect is on the person. And so I think a big piece of it is just informing people of all the different career paths, too, that are available. So I commend Aloni Las Positas for creating these spaces for us where we can get an opportunity to just share about the different career paths that we have. And we were talking before with a student about how so for me personally, I went to a four-year institution and happened to just fall into grad school, mainly because I couldn't find a job. And I had a research advisor who told me, she said, oh, you should continue doing school. Like, you're really good at research. You should do grad school. And I was like, oh, I don't know. Grad school just seems like, that seems like a PhD. Me, like, I don't come from fancy folk. Like, my mom was, because like my mom was a kindergarten teacher's aide and a nanny, and then my dad was a construction worker, so I thought, well, I mean, getting a bachelor's is good enough. Like, my parents didn't even do high school. I don't want to be fancy and get a PhD. And I also didn't see people that look like me, so I just never thought about like, oh, I could be in that role. So I just kind of stumbled along through undergrad and grad, and I didn't really even know what kind of a career was out there for me. So when I look at people like Sierra and Javier that have come to the lab at such an early stage in their education, I'm impressed that they were able to find out about these jobs, because I did not even know that the lab existed. So I've been at the lab for 4 years. I didn't know the lab existed 5 years ago. Like, that's how uninformed I was, because I think sometimes, especially at the four-year universities, a lot of the focus is on academics, and so you don't get a chance to take a step back and think, why am I doing all of this? What are the opportunities out there for me beyond what people think of like doctor, lawyer, right? So maybe I can also just turn the question over to Sierra and Javier, like, how have, being in the community college and doing internships help you to understand sort of like what your career goals and aspirations are? Because I know for me, I'm really impressed that, so I initially said Ohlone because I got Javier through Ohlone College. There was somebody who sent me his resume and said, I have a really bright student, and I looked at it and I was like, oh yeah, Javier seems like a perfect fit. But it's funny because he did a four-year university first before he went to Ohlone College, and it was Ohlone College that really helped him get to the real job aspect of it. So maybe I'll let Sierra take a stab at it.
So for me, it was 100% luck and this school. I grew up right next to the lab, and so I always knew about it, but I didn't know anyone that worked there. So when I started here, I was actually a psych major, and then a couple years in, I decided I wanted to switch to engineering. And Kelsey was the coordinator for the mechanical engineering technology program, and she saw that I was enrolling in all these engineering classes all of a sudden, and so she reached out to me and said, hey, I have this program, and it's really structured, and a part of it is that you get to interview for an internship at the lab. And I thought that was the coolest thing in the entire world. So she really helped me get my foot in the door there. I ended up doing the interview and getting the job, and then after the interview they actually asked me back, so that's kind of how I got in. But I would have never been able to really find it without the school's program and them reaching out to me and pretty much just saying, here you go, just do a good job. So
So selfishly, so what are your career aspirations? Are you thinking about staying at the lab or going on to school, or?
I want a bachelor's degree.
You do? Okay.
But the lab is great.
Yeah.
So hopefully I can do both of those at the same time. We'll
Yeah.
see.
So I definitely would say, like, you know, if you think something is cool, definitely try it out when you're young, because that's the best way to find out if you like something or not. For me, going through the grad school process was important because it taught me, like, oh yeah, I could do this. I could get a PhD. Just because I'm not this, you know, nerdy looking old dude doesn't mean I can't be an engineer. But you know what? I actually had to try it out because I honestly, like I said, I thought, like, I don't come from fancy folks. I don't need that kind of. But it wasn't until I did it where I was like, hey, this isn't, you know, this thing that I imagined. Like, I could do this. So definitely I encourage everybody to try internships and to definitely take advantage of the programs that Las Positas has to get you exposed to different careers and different career paths.
Okay, with that, what great research, wonderful stories. Let's make our speakers here. Next one, April 9. See you then.